Okay, so you can ask me questions. I look like a pirate mixed with like a trucker, but I can answer like some of them, alright? Like I understand that. So I'm in Dilson Rassia's laboratory. We study muscle biophysics. So I look at like molecular interactions more than like gross muscle structure. But I'll do my best to answer your questions. And I did my master's in kin as well. So I'm in physiology right now, so I'm a little bit of a sort of a wide background in these things. So I just added a quick overview. I don't know if you guys have these other slides that we uploaded them like really late this morning, but we'll go over entire muscle structure. Then we'll go through the hierarchy as you get more bundled into. You have bundles of cells, and then we'll go within those cells and look at the protein interactions. And the contractile units are called sarcomeres, so we'll go into the sarcomere composition. The sliding filament theory, so how these sarcomeres work to produce forces. The excitation contraction coupling, so how the signals are converted into mechanical forces, and the length tension relationships. So the length of the sarcomeres and how they produce forces, and then you graph those, and it's really important for, I guess, in the field. Okay, so we'll start with the hierarchy. So muscles are these bundles of bundled cells. So it's like, I guess I'll start sort of backwards at the bottom. These are the cells themselves, and they contain the contractile units. Those are in turn bundled and surrounded by fasciculi, and they each have their own surrounding membranes. So, and those are in turn separated by other membranes, and they're enclosed by an entire epimysium or fascia. So, the fascia is important because when you contract your muscle, you put a lot of forces on each muscle, and if you didn't have this, you'd sort of tear your muscles and blow them apart. So this keeps it from one, sort of, it keeps it together, and two, actually keeps the muscles smaller. So it's a good idea when you're exercising, a little bit of like applied stuff, stretch your muscles, and it actually stretches out your fascia, and you can actually slightly increase muscle growth. And so that's kind of an interesting fact. So I won't go too much into each of these, but you can just look at them just for your information here. So they're enclosed in each other, and then we're going to look more in detail at this level, and particularly the inside, the sarcoplasm, which is where all the stuff exists in the cell. All right. Okay. Great. So the part that we're going to be mostly looking at is the cell itself. So each cell has these networks around the contractile bundles, and they're called the sarcoplasmic reticula. And when you have a signal coming in from your motor neuron, it has to travel through somehow, right? So it travels through this network, and one of these, there's a groove along this network called the transverse tubule. So the transverse tubule is contiguous with the sarcoplasm, or like the muscle jelly, and once the signal passes through here, you have calcium, which is released from the SR, and in this region, if you look at the like electron micrographs or in microscope, they call this little region right between the sarcoplasmic reticulum and the transverse tubule the triad. And the reason why they call it triad is because when you cut the muscle, it looks like SR, transverse tubule, SR. It's not actually like a triad in and of itself, it's just like a relative term. So there are three things that flank each other, and that's why they call it a triad. And so we'll look more into how this actual contraction mechanism works. But what happens is once the SR releases calcium into the sarcolemma, it interacts with the contractile filaments, mainly tropomyosin, which allows myosin-actin binding interactions, which allows a power stroke or force generation. So we'll go more into how that force is generated. But these tubes are necessary for transferring the force really quickly across the muscle, sort of instantaneously, right? All right, so this is pretty simple. Muscles are composed of mostly water, most cells. Proteins, so we'll talk mostly about these two here, but titin is a really, really large, actually probably the largest protein in the human body. It's a passive element, and that means that when you stretch your muscle, there's a resistant force, and that is provided by this titin molecule. So as opposed to other resistive forces like, I don't know, like cartilage or something like that, which you'd imagine is also sort of has tension on the outside. This is actually within the muscle itself. So myosin is your main motor protein, so this is responsible for contraction, the actual mechanics, and it interacts with actin, and this is the substrate of myosin, or the filament that myosin pulls on to shorten which we'll talk about soon, the whole sarcomere. Tropomyosin regulates the interaction between actin and myosin, so when you have calcium present, it releases the inhibition on the actin binding site, and then these two things can interact. And myoglobin is just an oxygen binding protein. So like mammals with a lot of myoglobin can hold their breath for a long time because they're binding a lot of oxygen. Not too much to say about that in this lecture. Interesting fact, it was the first protein to be discovered with X-ray. Shown to like the protein composition with X-ray crystallography, and they got the Nobel Prize for that. So it's pretty important. And 5% of the muscle is composed of ATP. So basically energy molecules, calcium, second messengers. Signaling molecules, magnesium needed for ATP, and then these basically ions for a million different things. Amino acids, fats, glycogen, which we will not go into this lecture, but they're there and they're important. I can do that here. All right, so each fiber bundle is surrounded by blood supply, capillaries, and they run in parallel. So as your muscles contract, these things contract in turn. The actual vascularization changes when you do different types of exercise. So, or you get older, you get sick, and it's pretty important for, like in histology, when you want to take a look at what's going on, you can actually look at what happens to the blood vessels. So do a lot of exercise, these things become more branched, they go through the, so like in your marathon runners, you have more of these around the fibers. All right, and they're important for oxygen delivery, nutrients, CO2, and heat removal. I'm going kind of fast. I feel like I'm talking super fast. Just let me know. All right, so okay, we will now talk about the actual contraction, or the site of contraction, the sarcomere. So the sarcomere is where the actin, myosin proteins live, and they're placed in parallel like this along the length of the bundle, and so what Dennis actually made these. So what I think he's trying to say here is the more sarcomeres you have, the more total shortening you have, but it doesn't mean that the sarcomeres themselves, between each sarcomere, there's no change in shortening. So every sarcomere changes right, roughly speaking. They shorten at the same speed, the same amount. So if you have a longer muscle total, all right, you're going to have more total shortening, and eventually that translates to more higher velocity of shortening. But each sarcomere is like every other sarcomere. It's just the length of the actual muscle fiber might change. So in your deltoid, it's shorter than in your bicep, right? So your biceps will actually contract more quickly than your deltoids because they're longer muscles and there are more of them in parallel, okay? So how does this interaction work? How do the sarcomeres themselves behave? And the main players in contraction are the myosin motors, which we briefly talked about before. They respond to muscle activation, and they interact with actin filaments. So these are thin filaments, and the myosin are the thick filaments. And on the thin filaments, you have troponin complexes, which regulate interaction. So you need to have the calcium present to release inhibition. And when these things all work together, it shortens the entire sarcomere toward the center, away from the Z line toward the end. All right. Any questions?
What's the difference again between troponin and tropomyosin? Like tropomyosin, we use the calcium, right? With this troponin.
Yeah. So they are always together. So you have the actual filament. So you have the troponin. So you have the troponin complex, and then the tropomyosin filament. All right. So it's just the different names for the filament itself and like the little the complex on top. Okay. And I think there's like a little bit. So how does this regulation work? So the tropomyosin filament follows the alpha helix, or the structure of the actin filament, and on top of it, there's a troponin complex which is docked. And the troponin complex has three parts to it. You have troponin I, which it inhibits when there's no calcium present, and inhibits the interaction between myosin and actin. You have the calcium binding domain, troponin C, and then troponin T just binds troponin to tropomyosin. And you can experimentally isolate these two things. For like in our lab, we do a lot of like biophysical measurements on these things, so they're separate entities. And you can actually, so calcium comes in, removes the inhibition, and then opens up the binding sites. Okay. All right, so this is more just more like sarcomere anatomy. So we have each sarcomere extends from Z line to Z line, and this is like the functional unit, but you can think about it as like a half unit, so a half sarcomere. Between so where there's no myosin, it's called the I band, or when they looked at it a long time ago, they called it isotropic region because it just looks white and clear. And the Z line is where the actin filaments attach, and I think this is just more actin, just not the Z line is composed of more actin, just not in a contractile formation. As you go past the Z line toward the center of the sarcomere, you start seeing some myosin heads, and this is where the thick filament starts. And myosin heads, so you see here that they're all sort of like arrayed with each other. They spontaneously attach to each other, so they can act alone in solutions, for example. Experimentally they can act alone, but they spontaneously form these clusters. And so these clusters form the A band, so from one end of the sarcomere to the other end, and they attach to the center of the sarcomere with through an M band, and this is what limits the range sort of limits the range of the entire contraction of the sarcomere. In the middle of the sarcomere, you have so the A band is named because it's anisotropic, because it has both the actin and the myosin filaments together. So when you look at it, it's a little bit gray. So that's why they call it that. And then H zone, there's nothing in the middle, and it's completely clear. It's called, I guess, the main German word Helig, meaning good. I don't know, they're like clean things or something. And that's basically the entire structure of this. And it changes slightly in different fiber types. So in slower fibers, or like distance athletes, like the Z line will be a little bit larger, and then you have different types of myosin heads. So these things are a little bit adaptable. It takes like 30 days of like activation and different types of ways to change. Well, this is actually faster than that, but to change like your fiber type and all that stuff. But we don't really go into that this lecture. Any questions? Okay, so we're going to shift a little bit into like more the gross architecture again, and it's important because it has to do with the sarcomere, how sarcomeres work. So architecture is basically the fiber alignment relative to the force generating axis. So there are so I guess I didn't really provide I can't believe we used a chalkboard. But all right, so does everyone understand what a force generating axis is? Yeah, like roughly speaking. Okay, well then I won't go too much into it. So this is like a muscle fiber, and if these are the fibers, and then the sarcomeres are oriented like that. Okay, so which way is the force generating axis? Which way? Where's the force going? Up, exactly. So if I draw it like this, which way is the force generating axis? Really easy. Exactly. There you go. So, all right. So there's an angle here relative to where the fiber is oriented, and then that is the pennation angle when you're not localized. You're not located parallel to the force generating axis. Okay. So this pennation angle allows for more packing, and then different types of fibers will generate different types of forces based on this packing. All right. So pretty straightforward, right? Any questions? Okay. So, two important parameters affecting muscle function are physiological cross-sectional area and muscle fiber length. So, PCSA is the sum of all cross-sectional areas within the muscle, and it's proportional to maximum force. So, if I were to tell you that if I were to tell you that this is the cross-sectional area, you'd believe me, right? But for a pennated muscles with pennated angles, is this also cross-sectional area?
No.
Why?
It needs to be perpendicular to the fibers.
Yeah, so this is where the force faction is, right? So it's not actually the true cross-sectional area. So when you actually when you start having more of these things, did I draw anything weird? No. Okay, good. Just teachers always do that stuff. All right, so this is one of them, and then the next one is going to be like that. Okay. So this is the actual physiological cross-sectional area. If you just took a cross-sectional area like that, then that doesn't work, because it doesn't tell you exactly how the force is being generated or or the proportionality to the force. And the muscle fiber length has to do with how many sarcomeres exist in series. So I think we talk about this a little bit more. What we do? Okay, so it has to do with shortening velocity. So okay, this is fiber 1, fiber 2. Based on what we said, which one is going to contract? Which set of sarcomeres are going to contract faster?
The lower one.
All right. Do the sarcomeres actually? Do individual sarcomeres actually contract more slowly or more quickly than the other? No. Okay. So if you have a lot of friends and they're all sitting on rolly chairs and then they sort of pull, on each other, I get it. Look at my birthday parties. Then you don't actually. They all pull, like you have two lines of people and then they pull. The actual rate of contraction for each sarcomere is going to be the same. Now, these people that are pulling at the far ends are going to be moving with greater velocity because everyone's pulling together at the exact same time. We're assuming that the signal is dispersed along the fiber instantaneously, okay? Which roughly it is. So these ones are going to contract by this much, okay? And so this is sort of like an additive thing, and then all together, this is going to result in a greater contraction velocity. So that's basically what they mean by that. That's in essence what the length velocity relationship is, which we're going to look at next week. All right, make sense? Great. All right, so some of the advantages and disadvantages. So fusiform muscles, I guess I may as well should have said it a little bit. So there are different types of these parallel muscles. One of these types is called a fusiform muscle. There's also strap and fan shape. We're not going to go into the different types, but this is the one we're using right now, fusiform. So the biceps, the fibers are oriented parallel to the force generating axis, and so you can pack them into this area without any of them being cut off by the area, right? So the advantages of that are basically what I just outlined here. You have more sarcomeres in parallel, and the fibers are pretty long, so they can shorten with high velocity. And this is really good for good over the angle of flexion. You can do this quickly, and it's a long-acting lever arm. But the problem is you only can fit 4 fibers into this area, so you're going to have a lower physiological cross-sectional area, as opposed to something like a pennate muscle. So, like we sort of talked about over here, when you start to draw the cross-sectional area on this muscle, you're going to see that it goes down, up, and then you add these things together, and then you have a longer physiological cross-sectional area, right? So what that means is you can actually fit more. If you take the same area as we did over here, you're going to actually be able to fit slightly more sarcomeres in. So think of this in terms of sarcomeres and not fibers, because that's the actual functional unit, right? You're going to have more actionable units within this small area. Now the disadvantage of that is you have more of these guys. So if you start taking the average velocities of each of these fibers, this one has a slower velocity, this one has a slower velocity. Maybe these two have a similar velocity to these, but on average the population is going to have a lower velocity, okay? But these are stronger muscles, so they all have their own functionality. Questions? Good. Okay, so how does this sliding work? Basically, or how does this contraction work? So the shortening, so it goes from outside in. So Z lines sort of mesh together, or they start getting close together, and then the actin filaments touch, and in some cases, when you're really shortened, they overlap. So this will actually get even smaller than this, and it's limited by, roughly speaking, the length of the A band, or where the myosins exist, but even those can be compressed slightly. And this is all powered by ATP hydrolysis, which we'll talk a little bit more in the next slides. But again, this is just a really simple graphic showing you what the sliding looks like when we take it from that to this. Okay. So I guess this is kind of a crazy diagram showing you the entire process. The YouTube video doesn't really work, so I forgot to change it. Sorry about that. I have a different one. And, may as well show that instead, because of course, man, it's crap. Okay, well. Alright, well, I guess while that happens. Alright, well, yeah, it always has to happen during lecture like that, but so be it. Okay, so I'll just take you through this, and then I'll just link the video after, or if it ever loads, then I'll just play it at the end of the class. But basically, you have an action potential, which is initiated by your brain sends a signal through your motor neuron, and then you have this thing called the neuromuscular junction, and you have these acetylcholine vesicles at the end of your motor neuron, passes through a synapse. All right. This is all extra stuff. It's not part of, I guess, what he wants you to know, but we'll get into that important stuff. I'm just outlining how it works. Next, so this acetylcholine signal is integrated at the neuromuscular junction. Things happen, and the sarcoplasmic reticulum releases its calcium into the sarcoplasm. The calcium then interacts with the troponin-tropomyosin complex. Inhibition is released, and then what happens at this point is the muscle actually was being inhibited from acting on the tropomyosin, the actin by ATP. So you actually need to have energy present to prevent yourself from contracting. So, which is a little bit counterintuitive, but what happens is the ATP it has to be basically hydrolyzed or cleaved, and once you have hydrolysis, you can actually interact with the actin filament. And then when you release different parts of ADP phosphate, so you release the phosphate, it actually causes a shift in the myosin head or the motor, and then this is like it's a 45-degree shift, so it's like pulling on a rope. And this rope pull happens many times a second, or it's like 10 ATPs per second or something like that, and then you have like a really small change in conformation, and you step along the filament, like 7 to 10 nanometers. And so this time happens many times, and it's like pulling on a rope, and then the sarcomere shortens. And this happens over and over as long as you're contracting. And then when the cycle is done, everything is cleared, there's no more calcium, you stop the binding, and then ATP binds back onto the, so it's always binding, unbinding, there's no more stimulus for it to bind, and then it just sticks to the myosin, and then it's stuck like this, alright? So, like rigor mortis and things like that, there's no more ATP, or ATP levels start depleting, and you actually are contracted because you need energy to prevent constant contraction. So remember that. It's a little bit counterintuitive, but that's how it works. So now we're going to talk about what does the force look like at each length of the sarcomere. So what happens as this structure gets shorter, or in some cases longer, right? So the length of the sarcomere determines the force output of the entire fiber. And so how they test these things is that they put the muscle in isometric contraction, or the sarcomere in isometric contraction, so like this, or like that, and then they activate it. So activating it means you give it a stimulus to contract. But you do so, and you keep it at each length, and as you do that, you take the force measurements, and you correlate it to the length. Alright? And so you get this sort of curve. And so we're going to look at each part of these things, and we're going to look at why you have this much force, or tension, percent of maximum, versus lower or higher. So, at the top of the tension length curve, you have a plateau. And this is where the maximum overlap exists. And it's limited by the center portion here. So, I have a quick question. What would happen if you had more myosin heads right in the middle here? What would this, if it's possible magically, let's say, what would this graph look like? Just this top portion. So this is based on the total number of possible interactions. Let's say I increase the number of myosin heads just in the middle. You have more possible interactions. Okay? So I know it looks like there's a bare zone because it looks like it's like a flat number of possible interactions. But if you increase the number of myosin heads, you have a transient increase in the total number of interactions. So it'd be more like a peak at a certain point. But because it's limited by the M-band and the attachment, and because this is what determines your maximum force output, the total number of possible interactions, you plateau it at the top. Alright? So between 2.2 and 2.25 microns of sarcomere length, you're going to have the same force maximal interaction. Does that make sense? Yeah? Okay. So you're limited by this bare region. So now we're going to move to the left, so we're going to shorten the sarcomeres more. You actually start to lose force. So based on this, I don't know if you guys can see this super clearly, but look at where the actin is and look at where the myosin is, and look at the total number of possible interactions. Why does that explain this decrease in total tension? So I don't have like a guess. Wrong guesses get extra points in my book. I love wrong guesses. I do them all the time. Okay, so what happens is you have a little bit of overlap, which means what is this guy doing over here? This little actin piece is not exactly. So he can't interact. So the total number of interactions, right? So you actually have a decrease. So again, this is based on overlap. So all of this stuff we talked about, everything like all this pennation angle, all this stuff, it's based on the myosin, roughly speaking, myosin-actin overlap. So if you forget anything, just remember overlap, and then that tells you how much force is going on because these guys keep interacting, right? And the more possible interactions they have, the more force they can produce. As it shortens, you have more actin. The actin is inhibiting the other actin from acting on the myosin, and you only have a part of it which is able to generate force. And at really short lengths, the actual myosin starts getting compressed. I don't actually know what that means, but I guess it just means that it just doesn't work as well. But roughly speaking, the overlap is decreased. Now we start going to the other side. So when you start having longer sarcomeres, why does your force decrease? Based on this image, why are you getting less force?
Maybe because like even if the myosin is the same length, like it can't grab those ends of the actin.
Exactly. So you actually decrease overlap for a different reason. So now it can't reach. So each of these guys have these myosin heads has a slight reach, so they have a little bit of leeway. But once you start getting into these 3.5, I think it goes up to like 4 microns for a sarcomere, you start to lose force at this rate. So again, you're limited by your H band. You're not going to have any interaction in the middle to plateau, and then you start to lose the actual possible overlap, and that's just because you can't reach anymore. And actually, that is represented really nicely by this, and it's also in the next lecture. So this just sort of relates the overlap with how the maximum tension is generated along this curve. And it's since been changed a little bit in the most recent literature. There's like a few other phases, but for the sake of this course, this is a really nice outline of how this works, and I think it really ties in how all of this stuff works together. So don't think of them exactly, aside from like the muscle hierarchies, think of these things as really dependent on each other for an entire mechanical view of the system. And it all comes down to how these cross-bridge interactions work, and then you can think about velocity and shortening and force generation, and then you have a really nice picture of how muscles work in unison. Okay, and so if you guys have any questions, I don't think there's anything else. Okay, so that was basically the whole thing. Yeah. So any questions?
Can you go through that, like the velocity length image?
Sure. Alright, so I think we're gonna go more into this next week. I haven't looked over the lectures like too much, but so when we talk about shortening velocity, we talk about velocity of the entire fiber. Okay? So not each sarcomere. So each sarcomere, aside from like muscle fiber type differences, which I know it's not important in this lecture right now, we're going to assume that all sarcomeres shorten at the same velocity. So if this guy shortens with a certain amount, within a certain amount of time, okay? And then these guys all shorten within a certain amount of time. In the same amount of time, who's shortening more in total?
The longer.
Yeah. So if you look at the actual velocity, so the distance over time, okay, this guy covers more of a distance, okay? So just based on like really basic speed, this one is just going to shorten just because there are more sarcomeres in tandem. So this guy is shortening at the exact same time as this guy. And if you imagine, this goes really far out, and he contracts at this amount, he contracts this amount, and this one. In the same amount of time that these contract, this guy is also moving. So imagine being at the end of this chain, and how much faster are you moving than the ones, like you won't feel it if you're in here, but if you're in here, you're getting the forces from everyone else, right? You're feeling the velocity change. So that's why, I don't know if that makes sense, but, right? Okay? Any other, outstanding? Okay, so, yeah, so if you have any questions, just come talk to me later or send me an email. I don't know if I'm on the TA list or whatever, and I'll see you guys next week for the second part of the muscle. Thanks.
Turn this thing off. I don't know, is that like super speedy? Am I like super fast? Okay, well. Fantastic. Do you know how to turn this off? Oh my god.